Assalamualaikum and hi. My name is Siti Muzaifa binti Mursalin. My matrix number is S52103 and my supervisor is Prof. Madya Dr. Abu Hena Mustafa and I am from Faculty Fisheries and Food Science. My title is Decomposition Rate and Nature of Rhizophora Species as a Review. This is my contents. First is introductions, and next materials and method, result and discussion, conclusion and references. For the introduction, first I want to talk about mangroves. Mangroves are highly productive ecosystem in food chains that occur at the intertidal zone along the coastline, estuaries, and island in tropical and subtropical. Mangroves are very important in total organic carbon for trap, uptake, and recycle as suspended litter in the water column. Rhizophora species mostly can be found in the coastal habitat of the moist and tropical and subtropical. Only 8 species of Rhizophora species have described that are widespread in worlds. All Rhizophora species taxa have their own characteristic like large water biome, propagule, and ability for long distance dispersal. And then next is decomposition. The meaning of decomposition is the process in which organic substances are physically broken down and converted to chemical substances to produce carbon dioxide, water, and release energy. Decomposition of any biological materials is very important in nutrient cycling and reduces best for change within any ecosystem. Factors of decomposition on mangrove forests are influenced by several parameters such as species, seasons, and the position. Next is leaf litter can be defined as the major contributor to annual detritus production about 30% until 30% until 70%. Numerous factors including the environmental parameters is macrofauna and macrofauna are responsible for the composition of mangrove litter. Next is my problem statement to review the factors that cause the changes in decomposition of mangrove Rhizophora species. These are related to temperature, tidal inundations, seasons, wet and dry, rainfall, and climate zones. For the objective, to review and analyze the decomposition rate of mangrove Rhizophora species. It is my material methods. 3 methods is searching, screening, and data organized and reporting. For the searching, first the article and journal was searched at 4 different databases such as Google Scholar, Mendeley, ResearchGate, and Science Direct. The strategy by looking at the title with the keywords such as Rhizophora species, decomposition, and mangrove, and save in the file desktop. The compatible references from the article and journal were evaluated and help to gain more data for my project. All the article and journal was screening by the title, abstract, and the result for the identify and fulfill the inclusion criteria. The criteria for the review project were investigated the decomposition rate of Rhizophora species at different location and species. For the data organized reporting, the data organized arrange all data according to other names and year, Rhizophora species, location, and season. For about 20 article journals were chosen from the 50 article journals that I have. Prisma guidelines were using in this project for reported all the information. This is my results and discussion. This is the map about the global distribution of 8 species of Rhizophora species and the most found at tropical and subtropical. This is the Rhizophora mangle is the most dominant of species in the world because this species' ability to survive and diverse in climatic and environment. Next, this is the comparison of leaf decomposition rate about litter loss with different Rhizophora species with different place. This is I want to talk about the tropical and subtropical from the location. Next is the decomposition constant with different species of Rhizophora species and different location. This is calculated by day and trade white. My discussion is the rate of decomposition accelerate with the wetting and drying, and the optimum of decomposition was 70 of percent of moisture. During the wetter month in Malaysia, the leaf litter of Rhizophora apiculata degraded is the faster than the other species. And in Kenya, Rhizophora mucronata is higher rate of decomposition during the wet seasons. During the dry season in summer, the air is more humid and contain more water, thus can encourage the decomposition process to occur actively. In the Mata mangrove forest, Peninsular Malaysia, Rhizophora apiculata and Rhizophora mucronata is higher in the leaf litter decomposition. Leaf litter mass loss is faster during the dry season in tropical area because the drying rewetting cycle to encourage the decay process. My conclusion is generally higher and faster litter decomposition rate are observed in Rhizophora species, which is depend on season, location, and other climatic conditions in different ecoregions of the world. Rhizophora species 3 are developing the highest elevation species in specific zonation area of the intertidal zone. The leaf litter quality during the decomposition process are depend on the mangrove species and season and impact ecological functional. Rhizophora species decomposition product may be available for the mangrove and detritus consumers within the soil. Okay, this is my references. Thank you. That's all for me.